good morning, or good afternoon, or evening, wherever you are in the world. We are happy to host you today in the webinar of the International Society of Bone Morphometry. The International Society of Bone Morphometry is really a very nice place where anyone interested in all aspects of quantitative assessment of bone across scales. We have a lot of options and opportunity for leadership panels and early career opportunity. It doesn't matter what you are doing related to bone, you are always welcome. If you'd like to do like some clinical research, or if you are interested in spatial transcriptomics in skeletal tissue or imaging, we have different working groups, and all these working groups are trying to gain more expertise and knowledge and helping each other related to bone quantitative assessment. So this series are organized by the Early Career Investigator Committee, which is I'm honored to be one of them. And I would be happy to announce the next webinar, which is going to be actually in January. So on December, we're going to have an off. Everyone should celebrate the Christmas time. But on January, we'll be lucky to have Dr. Franco, and he will talk with us about tissue clearing and light tissue microscopy with musculoskeletal tissues. In addition, we'll be very happy to announce that Dr. Krista Mays going to be our keynote speaker in the ISBN meeting in 2026, which is going to be in London in July. And I think, if I'm right, Naomi, Dr. Krista was your PI in the PhD. So it's a really nice coincidence.
Yeah, that's correct. Yeah. Unfortunately, I cannot go because I have to go to a wedding then.
Oh, I'm sorry.
Yeah.
But it's nice. It's a good coincidence. So please put this in your calendar, and we'll be so happy to have you and seeing you on July 2026. Also, if you'd like to be updated with all news related to the society, please follow our page on LinkedIn. It's very active dynamics. We share our newsletter, our meetings, our announcement. So please follow our LinkedIn page. And finally, we would like to thank Alexion for supporting this series. So thank you, Alexion. And moving to our start today, I'm so happy to present to you Dr. Naomi Dirckx. And I would say she's an early career investigator, still she's young, but at the same time, she has a very big and very nice portfolio. She has been trained as a PhD student in Belgium under the mentorship of Dr. Krista Mays, as we mentioned. And then she moved to the US to do her postdoc with Dr. Thomas Clemens. And I think she moved with Dr. Clemens from Johns Hopkins to Maryland, Baltimore, and she got her K99 and transitioned from a postdoc to assistant professor here in WashU. We are lucky to have you here today, Naomi. I will end my screen share, and please take it from here, and we will be so excited to hear the story today, hopefully.
Yeah, thank you for that introduction. I'm very honored to be here and to present my work. So I will first start with a little bit of like history on bone cell metabolism. And then I will share some of the current literature, and then I will move on to what we are doing in our lab. So the study of bone cell metabolism already began in the early 1950s. Let me see if I can put the laser pointer. Yeah. Do you see my laser?
Yes, Naomi.
Yes.
Okay,
yes.
great. So the study of bone cell metabolism began in the early 1950s with William Neumann, who proposed that bone cells produce lactate and citrate, initially to help in like mineral mobilization and so on. And in those early tissue culture experiments, Neumann observed that bone fragments acidified their medium because it was turning orange. And that showed for the first time that bone was not metabolically inert, but actively performing glycolysis, even under aerobic conditions, which is similar to what we now know as the Warburg effect. But despite these findings, like in the next years, the focus shifted more towards like signaling in osteoblasts, like BMP signaling, Wnt signaling, rather than cellular metabolism, because people saw metabolism more as a background and not as a driver. And there were also some technical limitations because there were no direct tools to measure energy flux in small bone cell populations. So for nearly 30 years, bone cell bioenergetics kind of faded to the background of skeletal biology. But then in 2007, something happened. So in the lab of Carcenti, they discovered a new function of osteocalcin. So osteocalcin is secreted by osteoblast and can act as a hormone regulating insulin secretion and energy metabolism. And that reconnected the bone again to whole body metabolism. So it was kind of the metabolic renaissance. And at the same time, there were also more technological advances, such as the seahorse. There was mass spectrometry, 13C tracing, NMR, even later on mass spec imaging. So you could do a lot more to investigate osteoblast metabolism. So then in the next years, there was also a big wave of papers that came out about osteoblast metabolism. And they define important roles for glycolysis, glutamine, and fatty acid oxidation, and osteoblast differentiation and skeletal health. So metabolism was more and more recognized as instructive and not merely like supportive. So that was the birth of like osteometabolism as a field. And then since like 2010, 2014, we also started connecting cellular bioenergetics to the actual processes of mineralization, as they found that citrate, which is a TCA metabolite, was found embedded in the hydroxyapatite crystals of bone matrix, where it was presumed to define the bone material properties. And that's also a little bit what we are investigating. And so then we've come to a full circle, like 70 years after Neumann's orange medium. We're again looking again at lactate and citrate, but now with like more modern tools that allow us to see how metabolism shapes the mineralized matrix. So for a long time, bone was primarily viewed as a structural organ, but we now understand that the skeleton is deeply integrated into whole body energy metabolism, and it engages in a continuous two-way communication with peripheral organs. On the one hand, bone acts as an endocrine organ, as osteoblasts and osteocytes secrete molecules such as osteocalcin, sclerostin, and lipocalin, that regulate systemic processes from insulin sensitivity and glucose tolerance to like muscle function, fat metabolism, and even appetite. In my doctoral work, we also explored a non-endocrine way by which bone itself directly contributes to systemic glucose regulation. So by activating the hypoxia pathway in bone, we increase the glycolytic flux and glucose consumption by the skeleton, as you can see here on the PET. The red areas are very high glucose uptake. And this metabolic shift reduced the circulating glucose level in these mice and improved the glucose tolerance and lowered peripheral fat mass. And we also saw this clear inverse correlation between skeletal glucose uptake and blood glycemia. On the other hand, the influence also runs in the opposite direction, and metabolic diseases such as diabetes, obesity, and anorexia nervosa alter directly like bone remodeling and quality, while hormones and growth factors that modulate energy homeostasis like insulin and IGF-1, they can also directly affect osteoblast metabolism and bone formation dynamics. So these findings reveal that skeletal health and osteoblast metabolism and whole body metabolism are actually inseparably linked. And so a better understanding of osteoblast metabolism opens new possibilities for treating both metabolic disorders and bone fragility. So let's look first at how bone is formed and remodeled. So during bone formation, bone forms through two distinct processes: intramembranous ossification, where mesenchymal stem cells directly differentiate into osteoblasts to form the flat bones as the skull, and endochondral ossification, where a cartilage template is gradually replaced by bone for the formation of long bones. And bone isn't just a static structure that forms once in childhood and stays unchanged. Instead, it is a very dynamic tissue that undergoes continuous remodeling throughout life to maintain its strength and to adapt to mechanical needs. And this involves a delicate balance between osteoclasts and osteoblasts. In fact, the tissue that is resolved by osteoclast in 3 weeks needs 3 months to be deposited again by the osteoblast. So that means that the skeleton remains very metabolically active throughout life. And so it requires also a significant amount of energy, not just for the differentiation of osteoblast and for the proliferation, but also for the biosynthesis and mineralization of the bone matrix. So this continuous energy demand is often overlooked, but it's very fundamental to understand how bone maintains its health and function. So bone-forming osteoblasts originate from mesenchymal stem cells that can differentiate into myocytes, adipocytes, and of course, the osteoblasts. And once committed to the osteolineage, osteoprogenitor cells differentiate into mature osteoblasts that deposit the bone matrix. And after completion of bone formation, they differentiate into quiescent bone lining cells, or they go in apoptosis, or they become osteocyte, the mechanosensitive cells embedded in the bone matrix. So lots of studies on osteoblast metabolism, especially the early ones, have been done in vitro, which already gives us a lot of insights in their bioenergetics. But we have to keep in mind that the in vitro environment is very artificial and cells are substrate smart. So that means that they will use any kind of fuel available in the culture medium. So to truly understand how metabolic pathways shape bone formation, we started using genetic mouse models by using specific Cre drivers such as Ostrix Cre for early osteoblast or DMP1 Cre for osteocytes. So we can delete or modify specific metabolic genes at different stages of differentiation. And thanks to these mouse models, we've identified a lot of critical metabolic pathways and signaling molecules, as you see here, that control osteoblast function and bone formation. And in short, by studying these in vivo systems, we gain a deeper understanding of how bone metabolism is regulated and how we might target it to treat skeletal disorders. There are three key metabolic pathways in osteoblast: glucose metabolism, fatty acid oxidation, and glutamine metabolism. And I'll give you a quick overview on some of the literature. Unfortunately, I cannot go over everything because I would need like 10 hours and even more. So I'll do just a quick and dirty overview, so to say. Glucose is the principal fuel for osteoblast, I would say, and it plays a bigger role than just energy generation. So glucose is taken up primarily through GLUT1 in osteoblast. And during glycolysis, glucose is broken down into two molecules of pyruvate that can either become converted into lactate or fed into the TCA cycle in the form of acetyl-CoA for further metabolic processing. However, even in the presence of oxygen, osteoblasts metabolize most of their glucose to lactate, showing a strong preference for aerobic glycolysis or a Warburg-like metabolism. So glycolysis is not only for the generation of ATP, but it's also very quick, and it allows the channeling of glycolytic intermediates into the pentose phosphate or the serine glycine branches that supply either the nucleotides for proliferation, but also amino acids needed for one carbon metabolism and epigenetics, or the biosynthesis, such as collagen, for example. So the advantages of glycolysis is that it's quick and flexible, it gives a lot of biosynthetic precursors, and it is also maintaining the redox balance. Then the TCA cycle itself serves as an ATP generating hub via OXPHOS, but is also a source of citrate and other intermediates needed for lipid synthesis, histone acetylation, and amino acids and NADH. So Anjonia Günther and his colleagues examined how the balance between glycolysis and oxidative phosphorylation shifts during osteoblast differentiation. And for this, they used primary calvarial cells cultured for 7, 14, and 21 days. So he measured the oxygen consumption rate at 14 days and 21 days, and the extracellular acidification rate as a measure for glycolysis at these same time points. And he sees that with differentiation, there is a bigger reliance on both oxygen consumption rate and on glycolysis. But to see what they preferred more, he divided the oxygen consumption by the extracellular acidification rate. And then you can see in the later differentiation stages that osteoblasts actually rely more on glycolysis for ATP than on oxygen consumption. While at day 7, they both use it equally. So glucose metabolism is, of course, tightly regulated by the classical osteoanabolic signaling pathways. So both Wnt signaling and PTH stimulate glycolysis via the mTOR pathway that induces protein synthesis of glycolytic enzymes. Insulin signaling stimulates glycolysis by improving glucose uptake. Notch signaling, in contrast, actually suppresses osteoblastogenesis by downregulating glycolytic enzymes and glucose transporter, so it's acting as a metabolic brake. And HIF stabilization promotes glycolytic flux and supports also bone formation, as we saw from a lot of gain-of-function models. So in addition to glucose, fatty acids are also a major source for osteoblasts. Fatty acid metabolism, or beta oxidation, occurs in the mitochondria, and long-chain fatty acids are first activated to fatty acyl-CoA and then transported into the mitochondrial matrix through CPT1 and CPT2. And there, each cycle of beta oxidation shortens the chain by 2 carbons, producing acetyl-CoA, which is then fed into the TCA cycle. And then it generates energy through oxphos again. So compared with glycolysis, fatty acid oxidation produces a lot more ATP per molecule of substrate, but it's a lot slower. And in vivo studies have shown that inhibiting mitochondrial fatty acid oxidation, for example, through conditional deletion of CPT2, results in defective bone formation and reduced bone mass, confirming that lipid oxidation is required for normal bone acquisition. And then one mechanism for the regulation of fatty acids oxidation is Wnt LRP5 signaling. So this signaling pathway is classically known for regulating bone mass, but this paper also showed that it controls bone mass through osteoblast metabolism, and specifically fatty acid oxidation. So in LRP5 knockout mice, the authors saw the expected low bone mass phenotype, but they also uncovered a striking metabolic defect. Osteoblasts had reduced expression of key fatty acid oxidation enzymes, and a marked drop in fatty acid oxidation capacity. And because the osteoblasts were not using fatty acid efficiently, the animals accumulated increased body fat and had elevated triglycerides and free fatty acids. In the LRP5 gain of function model, they actually saw the complete opposite phenotype. So this study demonstrated that Wnt LRP5 signaling regulates fatty acid oxidation genes in osteoblasts, which is crucial for matrix production and bone formation. So beyond glucose and fatty acids, there is also amino acid metabolism. And osteoblasts depend on both the branched chain amino acids as well as glutamine, and they together provide the carbon and nitrogen needed for biosynthesis and to maintain mitochondrial energy and redox balance. So glutamine metabolism is particularly important. Glutamine is imported through SLC1A5 and converted into glutamate by GLS. So it can go then to two major fates. It can either be converted into alpha-ketoglutarate, where it can be fed into the TCA cycle, which is also called anaplerosis. And in addition to that, it can also be involved in redox balance by its conversion into glutathione, which is a key antioxidant that protects osteoblasts from reactive oxygen species generated during high mitochondrial activity. And this has been shown in this publication, where the authors deleted GLS in osteoprogenitors. And these GLS deficient mice developed reduced trabecular and cortical bone mass, as you can see here, due to the loss of glutamine flux into the TCA cycle and into amino acids, nucleotide, and glutathione synthesis. And as a result, osteoprogenitors had defective protein and collagen synthesis, as you can see here in gray. But that could be fully rescued by alpha-ketoglutarate supplementation. And because glutamine also supplies glutathione, GLS depletion increased ROS production and reduce the cell viability, as you can see in gray. But then this could also be reversed by supplementing the cells with a glutathione ethyl ester, as you can see here in orange. And finally, the study also shows that the anabolic effect of PTH increases the glutamine uptake, and that GLS deletion almost completely blunts the bone anabolic response of PTH signaling. So that's quite striking. So far, we have really scratched the surface of osteoblast metabolism, and that they rely well on glycolysis, fatty acid oxidation, and amino acid metabolism to generate energy, and biosynthetic precursors for collagen production and so on. But these pathways have learned us a lot, but they really don't fully explain how they make the bone unique as a tissue. Because unlike soft tissues, bone is mineralized. So we then asked, are there any metabolic intermediates that actually can assist in the mineralization process? And for this, we go back in time again. So in 1941, Frank Dickens, so that was already before William Newman that I said in the beginning. So he analyzed bone tissue using very old chemistry methods, and he found that the concentrations of citrate in bone were really high, like 70% of total body citrate was stored in the skeleton. And like I mentioned in the intro, it was first thought to acidify minerals and help in ion mobilization even before the osteoblast was identified. But despite the intersection of citrate and all the metabolic pathways, the role of citrate in bone biology was largely ignored for decades, apart from just being a TCA metabolite. So the real change in interest in citrate metabolism in bone started when these two papers using sophisticated methods such as solid state NMR and X-ray diffraction came out. And they both aim to model the structural role of citrate in bone. And they proposed actually that citrate is an integral part of the bone mineral, as well its degree of incorporation in the bone mineral as its spatial orientation in the mineral structure could play a role in maintaining the biomechanical properties of the bone, which is stability, strength, and resistance to fracture. So these papers made us actually very curious in citrate. And so we wanted to investigate how citrate metabolism works in osteoblasts and how it contributes to skeletal mineralization. So to start investigating this, we knew that citrate uptake from the extracellular space cannot happen by simple diffusion through the membrane. It requires a specific transporter protein. And one of the citrate transporters that gained a lot of attention is SLC13A5. And this is the only outside-in carrier that prefers to transport citrate over other carboxylates within this family. And the reason why it got so much attention is because it was identified as a longevity gene. So first in Drosophila and later on in C. elegans. And that's why its alternative name is INDI, which stands for I'm not dead yet. And more recently in mice, it was also shown that INDI knockout mimics caloric restriction in the liver and provides resistance against everything bad that is caused by a high-fat diet. So it looks like it's an interesting target to further investigate. And then indeed, we found it was very highly expressed in bone, even higher than the brain, the kidney, and the liver, and similar as expression levels in the testes, which are all organs that are very important in citrate metabolism. And to know where exactly it is expressed, we performed a pseudotime analysis of osteogenic differentiation from a single cell sequencing data set. And it showed that SLC13A5 expression was enriched in the late mineralizing osteoblasts and osteocytes and clustered together with other mineralization genes. In parallel, the uptake of radioactive labeled citrate increased fourfold over the course of differentiation of osteoblasts. And then if we delete SLC13A5 here in red, or when we treat osteoblasts with its inhibitor, that almost completely abolishes the 14C citrate uptake in the osteoblast. And if you look at spatial metabolomics data in vivo, where mice were injected with heavy labeled citrate, we also see that the knockout mice almost don't take up any citrate, while the control mice take up a lot of labeled citrate. So then we wonder, what is its role in mineralization? So for this, we used wild-type calvarial osteoblasts that were treated with the inhibitor or vehicle, or control and SLC13A5 knockout osteoblast cultures. And so detailed imaging of the calcium minerals showed a defect in the mineral nucleation in both the inhibitor treated as well as in the knockout versus their controls. So it's like a little bit more diffuse mineralization versus the very dense nodules that you can see in the control. And this phenotype could not be explained by alterations in collagen or calcium, nor by defects in endogenous differentiation capacity. But since we saw in the introduction that citrate is assumed to play a role in mineral structure, we were interested in the citrate content of the mineral matrix. So therefore, we designed a protocol to separate the mineral matrix from the cellular fraction in 2-week differentiated osteoblast cultures, and we treated them with hydroxychloride and methanol, respectively, to isolate citrate. And surprisingly, and opposite to what we were expecting, we observed a drastic increase in both the mineral and the intracellular citrate content in the mutant cultures. So either we mixed up the samples because we knock out the transport, how can there be more citrate in the minerals? Or there is a mitochondrial compensation due to the loss of citrate uptake. So to investigate the latter, we differentiated osteoblast again in the presence of 13C labeled glucose that is eventually converted into citrate in the TCA cycle with 2 labeled carbons. So mass spectrometry on the lysates of the mineral matrix showed indeed that increased citrate in the mutant cultures was mainly derived from glucose and generated in the TCA cycle. And also increasing doses of the inhibitor caused a dose-dependent effect on 13C citrate incorporation in the mineral. And in agreement with these findings that more citrate was produced from glucose, we found that SLC13A5 deficient or inhibitor-treated osteoblasts show a significant increase in glucose uptake from the medium. So to see what is happening inside the cell and how metabolites could be affected by this massive citrate capillosis, because citrate is involved in so many pathways. We did again intracellular metabolic flux analysis, but this time we analyzed the intracellular fraction. And of the many pathways affected, I'm showing here that there is increased citrate production to the expense of other TCA metabolites, as well as other anaplerotic amino acids. And in line with this increased glucose consumption and citrate cataplerosis, we observed a reduced in overall oxygen consumption by seahorse extracellular flux analysis, but also an increased ratio of ADP over ATP and phospho-AMPK, and a reduction in ATP production. So these osteoblasts were in an energy deficit because they just use everything to produce citrate and secrete that into the mineral. So as an intermediate conclusion, we can say that SLC13A5 is the main transporter for citrate uptake. And when you block it, you reduce citrate uptake, and you cause, as a consequence, a compensational mechanism in the mitochondria where they're going to produce more citrate. So what does that have, how does that affect the minerals in vivo? So for this, we look at 2 time points in young mice and humans, as well as in old mice and old humans. So let's start first with the young mice. For this, we look in to osteoblast full knockout and osteoblast global knockout mice of SLC13A5, sorry, and osteoblast-specific SLC13A5 knockout mice using the osteocalcium driver, because we saw that SLC13A5 is mostly expressed in these late mature osteoblasts. And we'll look at their bones and their teeth. So before we check the morphology, we just wanted to check if the mechanism that we found in vitro of increased TCA-derived citrate deposition in the minerals also occurred in vivo. So we injected young 5-week-old mice, control and knockout mice with 13C glucose for 1 week, and then we performed solid-state NMR that allows citrate identification in bone chips by 3 specific spectral bands. And indeed, as you can see here in this magnification, the NMR revealed a 2-fold increase in citrate in the bone minerals, which was in line with our in vitro findings. And this was also true for the skeletal specific knockout mouse. So let's see how does it affect the mineral tissue formation. If we look at micro-CT analysis, we see both in the global and in the bone-specific knockout mouse model that the cortical thickness was significantly reduced, and also the tissue mineral density in both models was reduced. If we do mechanical testing using the 3-point bending test, you can appreciate from these force displacement curves that both knockout models fractured sooner than their control littermates. So the bones were more fragile. Another interesting fact was that these mice had very wide teeth compared to their control littermates. And when we analyzed those teeth with micro-CT, we actually saw that this was due to a complete loss of enamel, which is the most densely mineralized tissue that we have. And with further 3D segmentation, we also found significant reductions in dentin volume and density, and an increase in pulp volume. If we look at the bone-specific knockout, we see a similar phenotype for the dentin and the pulp, but not in the enamel, and that is simply because osteoblast is not expressed in the ameloblasts. So to unravel the underlying mechanism of this developmental tooth defect, we investigated the teeth in our mouse model and found similar expression levels of SLC13A5 mRNA in teeth versus bone. And on immunohistochemistry from tooth sections from 2-week-old mice, we showed that there was very strong SLC13A5 immunoreactivity in enamel secreting ameloblasts and in dentin secreting odontoblasts. And solid state NMR again confirmed the increase in citrate that we found in bone, but even maybe more pronounced in the teeth. So then let's see in the human situation if there is any information on SLC13A5. So in humans, there are naturally occurring mutations that induce loss of function of SLC13A5, and this is a very rare disease, mostly characterized by epilepsy shortly after birth, but they have a lot of problems and intellectual disability, weak muscle tone, and so on. The clinical manifestations can be mild to very severe, but all the children that they have examined so far present with this tooth hypodontia and tooth hypoplasia. And strikingly, if we look at their micro-CT, because we were very lucky to get some primary teeth from these patients, so micro-CT analysis showed very striking similarities with our mouse model. So the enamel was very, very thick. It was not absent, just very, very thin. The density was also reduced, and also the dentin thickness and density was significantly reduced. By our collaboration with the Test Research Foundation, we also were able to get iPSCs, because we always get the question, do these patients also have bone mineralization defects? But since it's a very rare disease and they have so many more problems going on, they never did DEXA scans. They don't have spontaneous fractures, but they do show like the late growth. So with the help of Pamela Ruby, we have managed to differentiate those iPSCs from patients into lateral plate, mesoderm, neural crest progenitors, and then continue into the osteogenic lineage. And we've done that so far very successful here at WashU with the human tissue and organoid core. If you look at their mineralization capacity and we compare patient iPSCs with their healthy siblings, you can clearly see that patient iPSCs fail to mineralize. If you look at a patient line that failed to mineralize, and we reintroduce the wild type gene again of SLC13A5, which is called an isogenic control, we can see that mineralization defect is actually rescued. And if we do again our mass spec analysis on the minerals, we can see again that glucose-derived citrate is increased in those minerals, so could again be the cause for that impaired mineralization. So blocking SLC13A5 in young mice or human is bad for mineralic tissue formation. But how is this regulated? I'm not going to show all the data, but what we found so far is that cytosolic citrate is an allosteric inhibitor of glycolysis. So blocking SLC13A5 gives a dip in cytosolic citrate levels, releasing that brake on glycolysis. And then pyruvate is fed into the TCA cycle through the mitochondrial pyruvate carrier. However, to get citrate out of the TCA cycle, we need somehow a break on the TCA cycle. And so one plausible mechanism for enhanced mitochondrial secretion of citrate has been described by the lab of Costello. And this mechanism was first discovered in prostate epithelial cells and involves an upregulation of the zinc transporter CYP1. So this involves a zinc-dependent inhibition of amazonitase, which is the enzyme that converts citrate into isocitrate. So blocking this enzyme would truncate the TCA cycle and leads to citrate accumulation and secretion. So we found a significant upregulation of ZIP1 in SLC13A5 knockout bones, a significant uptake of zinc in these mutant cells, and a reduction of amazonitase protein. Moreover, metabolic flux analysis shows that the increased mitochondrial citrate production as a result of treating the osteoblast with the inhibitor was reduced when ZIP1 was knocked out. So it's a partial rescue. So to test the potential interaction between SLC13A5 and ZIP1 in vivo, we created a double knockout mice. So if our hypothesis is true, normalization of mineral citrate levels by ZIP1 deletion would then rescue the skeletal phenotype of SLC13A5 knockout mice. So solid state NMR experiments showed that citrate levels of ZIP1 knockout mice were similar to the wild type, but when we delete ZIP1 in the SLC13A5 null background, we see a little bit of an improvement of the elevated citrate levels. It's not a lot, but despite that modest change, it was sufficient to normalize the elevated serum citrate that we see in the knockout mice. And also the reduced cortical thickness that we see in the single knockout was normalized in the double knockout. If we then look at the bone strength, we see again that these single SLC13A5 knockout mice have an increased fragility that was again normalized in the double knockout mice. So in summary, we hypothesize that SLC13A5 is some kind of a citrate sensor that is required for osteogenic citrate uptake. And in normal physiological conditions, we believe that the extracellular citrate is deposited directly into the bone minerals. And when SLC13A5 is blocked, the mitochondrial citrate secretion is enhanced through ZIP1 upregulation, and elevated mineral citrate levels impair mineral tissue formation during growth. But what happens with aging? So again, we are going to look at a mouse model and in the human situation. So when we age, a lot of people have to deal with osteoporosis, and this is often very underestimated as a public health problem. So in particular, hip fractures are the most devastating of all the fractures, and that result in a mortality rate of 20% within the first year after fracture. So this is almost similar to some type of cancer. The figure on the right is another study that shows the probability of mortality for a period of 10 years following the hip fracture occurrence. So the chances of dying after a hip fracture in the first 90 days is around 16% of women and 25% of men. So that's quite high. And we already have a lot of anti-resorptive and anabolic therapies on the market that work well, but they all have their limitations, their side effects. And what is common in all of them is that they are focused on bone mass rather than bone quality. So we believe with citrate, we can really focus on that actual quality of the bone rather than the mass. And so a recent study showed that both mineral and serum citrate levels were reduced in a mouse model of OVX, so in the mouse model of postmenopausal osteoporosis. And these authors also found a positive correlation between plasma citrate levels and lumbar spine and hip bone mineral density. So that makes you wonder, can we reduce age-related bone fragility by elevating mineral citrate levels? And you're probably wondering, in all these previous slides, you said that elevated mineral citrate levels are bad, and now you want to go elevate them to rescue bone fragility. How does that work? So if we look back in the literature, we find a lot of positive associations of SLC13A5 with aging. For example, deletion of SLC13A5 reduces the blood pressure. It mimics the caloric restriction. In lower animal models, it extends lifespan. So we actually hypothesize maybe there is differential effect of SLC13A5 deletion on bone mineralization and strength in young versus old. And so that's why we went back to our skeletal specific mouse model with the osteocalcin-3 driver to look at aging. And so we looked at 1-year-old mice, and I know that is not considered aged, but it was enough to already see like an age-related drop in bone volume. And again, we saw an increase in citrate levels in these knockout mice. And so if you compare again with the 6-week-old, as a little recap, the 6-week-old mice had a reduction in cortical thickness. At 1-year-old, this reduction is not there anymore. At 6-week-old, they were more fragile, but at 1-year-old, that fragility is reversed and the elasticity is improved. At 10-week-old male mice, we don't really see any changes in the cortical thickness, but at 1-year-old, the bones became like very much wider. And that also caused, when we do 3-point bending, that these bones were like a lot stronger. So while deleting SLC13A5 is detrimental for bone formation and quality during growth, our data suggests that actually old mice with SLC13A5 deletion have more favorable bone mechanical properties compared to their age-matched controls. So could we consider SLC13A5 as a target for osteoporosis treatment in humans? And for this, we have used UK Biobank data. So UK Biobank is a prospective cohort study with deep genetic and phenotypic data collected on approximately 500,000 individuals across the United Kingdom between the age of 40 and 69. A rich variety of phenotypic and health-related information is available on each participant, including biological measurements, lifestyle indicators, biomarkers in blood and urine, imaging of the body and the brain. And also genome-wide genotyping data have been collected on all participants, and that provides many opportunities for the discovery of new genetic associations and the genetic basis of complex diseases. So to go a little further than genome association studies, we used Mendelian randomization to find causal effects. So in observational studies, it's often hard to tell something about causal effects due to the confounding factors. But Mendelian randomization uses genetic variants as instruments to study the effects of exposures on the outcomes in the presence of confounding factors. So Mendelian randomization basically mimics a randomized control trial where genetic variants are randomly segregated during meiosis in the population. And if you divide that population in affected allele versus control, and you keep the confounding factors equal between the groups, you can compare the outcome between those groups and do a risk assessment and actually predict a causal effect, so not just an association. So in our case, our instruments are SNPs in SLC13A5, our exposure is plasma citrate, and our outcome is osteoporosis risk. And intriguingly, we observed that SNPs associated with reduced function of SLC13A5 increased the serum levels of citrate, and they were also associated with a lower risk of osteoporosis in these biobank participants. While if we just look at high citrate levels in the plasma, we did not find that association. So you really need that reduced function of SLC13A5 to get this. So as a conclusion for this, we can say that the elevation of mineral citrate levels during aging actually could improve mineral tissue properties. And so why do we see those differential effects? So the current working model of how citrate works during mineralization is illustrated here. In the initial phase of mineralization, citrate acts as a nucleation factor, so it collects all the calcium phosphates and initiates the crystal growth. But in the later phases, it actually acts as a capping agent. And so with this, it blocks the crystal thickening but it allows the longitudinal growth. And that's how you get a typical plate-like morphology of apatite crystals in bone. And so our current hypothesis is based also on literature is that when you age, your crystals become bigger, and also the crystal, the heterogeneity of the crystal shape becomes lower. So you get bigger crystals and you get less citrate in between those crystals. So we think that when we delete SLC13A5 in the young age, adding that extra citrate really makes your crystals like very brittle and almost demineralizes them with adding more and more citrate. While if you add more citrate to like the very big aged crystals, you can actually reduce them to a more youthful morphology. So that's why we think that it could be more beneficial in aging to block SLC13A5. But we are currently investigating this. So hopefully, maybe next year, I can tell you more about this. And with this, I would like to thank a lot of people that helped to accomplish this work. My PhD lab with Krista Maas, because I showed data from there as well. My postdoc time in the Clemens lab and in the Riddle lab, and then now also my own lab and all the collaborators. So I'm very happy to answer all your questions now.
Thank you, Naomi, for this beautiful structured presentation.
Thank
Your
you.
insight into how osteoblasts integrate metabolic pathway highlights the importance of metabolic complexity for bone health. And personally, like your findings connecting these pathways to human disease condition were also very impressive.
Thank you.
And thank you as well to everyone joining us today. We will now open the discussion. So first is from the chat, Professor Hernandez. So wonderful talk. So your findings show changes in bone volume as well as changes in citrate content within the matrix. The biomechanical data you showed was for the whole bone and did not separate the effect of cortical thickness geometry from that of matrix alone. Without such data, all your biomechanical result could be from geometry, not a change in mineralized tissue as
Yeah.
you propose.
Yeah, that's completely true. But we also did stress strain measurements, taking into account this difference in the structure, and we see the same thing. So they're still more fragile if you look at stress and strain. I just didn't incorporate it in the presentation.
Good. And I also have one more question from Towser. Thank you for this wonderful presentation. I was wondering what are the effects of citrate on bone resorbing osteoclasts once they resorb bone? How do osteoblasts respond to different extracellular condition of citrate around them?
Yeah, these are very interesting questions. So we don't really saw differences in osteoclastic bone resorption in our bones. Like if we look at typical resorption markers such as CTX in the serum, we don't see differences. We also looked for the typical gene expression profiles, also rank ligand, OPG. Those were all like not very different. So I don't think there is an effect on bone remodeling. And we also didn't find the expression of SLC13A5 on osteoclasts. I don't think there is a direct effect, but it could be like one of the reasons why we say that differential effect could be that, for example, bone that has more citrate is somehow harder to resorb by osteoblast at the aged mice. That's why we see that improvement. We don't know that yet, but we are all like investigating that at the moment in order to like have a question to that answer. How do osteoblasts respond to different extracellular concentration of citrate around them? So in vivo, I don't know. But in vitro, if you add a lot of citrate to the culture medium, you actually inhibit the differentiation. And I think it is because they take up so much citrate that they start like just to demineralize the matrix or the calcium that they just deposited. Because I mean, citrate is something funny. You need it in your bones to have mineralization, but if you have too much, it actually just solubilizes the calcium precipitates. Because, for example, if you have kidney stones, citrate is used as a treatment to dissolve those stones. So if you have a lot of citrate, you just dissolve mineral.
Okay. So we have a lot of questions now. And the next question is like a wonderful talk. What are the parameters to say good bone and bad bone? I also like feel about
Yeah,
this one.
that's always an interesting question. I mean, especially here, because what is a good bone and what is a bad bone? We always think like, yeah, of course, if it breaks faster with load, then it's probably bad quality bone. But there is more like a nuance to this, I feel, because what we see like in our older mice, so the mice that we looked at, they were mutant from birth, of course. So they have to catch up first before they can become better. So next studies with just using the inhibitor in older mice or using the tamoxifen-induced Cree, I expect that if you use this kind of model, that you will actually see an improvement. Um, but if we look at for example, our females, they become a lot more elastic, but they don't break faster. So is that good or bad? I don't know. I think it's good because the bones are more like flexible to to like the load that you put on them, but they don't break. And I think that's what we want, right? We don't want to break our bones. So yeah, sometimes I struggle with this too. Like, how do you define like this is good bone or bad bone? But yeah, I guess the ultimate answer is probably like how soon it breaks.
Yeah, I agree. And next question is like, I was wondering, given how hypoxic bone is in terms of your cell culture experiment, did you ever account for hypoxia using a hypoxic chamber? Also, did you ever trace the expression of HIF1 alpha and was it linked to citrate?
Mm-hmm. Yeah, that's a good question. So mostly bone is considered to be hypoxic. Um, I somehow agree with that because it's probably more hypoxic than any other soft tissue, but it is also not hypoxic in a way that it doesn't have any oxygen, right? Because in all our models of HIF stabilization, we see a very dramatic phenotype. So if it would already be very hypoxic, we already would have like this default HIF stabilization, and more HIF stabilization wouldn't give you an extra phenotype. So I think the osteoblasts definitely have oxygen. So it is less oxygen than another tissue, but they still have oxygen. And so I worked in my PhD a lot with hypoxia. But back in the days, I didn't look at citrate yet, of course. But in I mean, if you really work like in hypoxia, I feel like that has a negative effect on osteoblast differentiation. But back in the day, I never checked if this was just because of citrate or for like many other things. So I have to look at this. It's actually a very interesting question.
Right, right. So now it's like a more like a hypoxia condition is getting like a notice in our societies. I also like a very agree about this question. And there's like a two question about like heterogeneity of the cells. Like first is like from Mohamedo, and
Mm.
with your dental findings, do you think odontoblast and ameloblast follow same
Oh.
mechanism as osteoblast, and what about them in your aging model as well? It's a little bit like a hard question, I feel.
Yeah, I mean, I think they do the similar mechanism because we saw also this upregulation of ZIP1 and SLC 25A1, which is the mitochondrial citrate transporter, the export citrate. So I would assume it is the same mechanism. But there's probably some additional things too. So one of the hypotheses is, of course, like since enamel is so densely mineralized, like adding that extra citrate is probably just demineralizing it completely. But also during the development of enamel, like certain levels of pH are important and they change also over the time of enamel formation. And citrate is also very involved in like acid base balance, so it changes the pH. So that could also be a reason why there is a complete absence of enamel in our mouse model. So it's probably just more than the mechanism, but I think that same mechanism is also in play in in the ameloblast and the odontoblast. But we haven't really investigated that into too much depth. Partially because there is not really like good models for odontoblast and ameloblast in vitro to like really do like the metabolic fluxes and so on. So if you have an idea and you want to collaborate on that, I'm totally happy to do that.
Okay, you need to collaborate with Muhammad.
Yeah.
And the next question is like might be more easier, like from Professor Hernandez. I noticed your mouse model altered osteoblast as well as osteocyte. Have you thought about the different effect of those two type of cells?
Mm-hmm. Yeah, that's an interesting question. Yeah, so it's probably both important in both, but in the osteoblast, of course, you have the effect of the mineralization. What it does in osteocytes, I don't know at this moment. But we do have a feeling that it is probably involved in this osteocytic osteolysis. But we don't have any data on that yet. That's just an hypothesis.
Okay, cool. And next question is like a great talk. Just wondering how we can translate bone metabolism in type 2 diabetes. So the question is, does bone may affect on type 2 diabetes or other bone-related disease?
I'm not sure if I 100% get that question. Do you think do you mean like can we
I think like
improve the
in
quality?
general,
Yeah.
in general, more like a pathological condition, like
Mm.
how they change the effect, especially for this type 2 diabetes, I guess.
Yeah, so type 2 diabetes is definitely not good for bones. So what we typically see there is that type 2 diabetes is not necessarily like affecting the bone mass, but also the quality, because often these patients, they don't have like bone loss, but they do fracture a lot more.
Right.
So it's definitely like the quality. But I haven't investigated if citrate plays a role there. I do know that when we compare the expression levels of SLC13A5 in um, low-fat diet mice versus high-fat diet mice, that SLC13A5 is differentially expressed. But that's just high-fat diet versus low-fat diet. That's not, I wouldn't say that's necessarily like type 2 diabetes, but there's probably a link there, but at this moment, I don't have any data to support that.
Got it. So like related to the pathological condition, like I do have a one question, like a point, like have you ever investigate about the injury condition about your citrate metabolism? It might be like something interesting for me as well.
Yeah, no, for sure. So I currently have a very motivated medical student who also asked herself that question. So I just got like my IACUC approval to do some fracture studies. So, and
Cool.
I think there's definitely going to be something because we're also investigating the role of SLC13A5 in cartilage. And we see that it has a very important role there in like biosynthesis as well. So in generating the precursors for glycosaminoglycans. And since like if you do like the endochondral fracture healing, it both goes through like this cartilage template and then it becomes mineralized. So I do think we will see some effects there, but we have not initiated that yet. But it's coming up.
Yeah, that's cool. And maybe final question is like, I'm curious how many mitochondria does an osteoblast and does the number change with age and hence alter mitochondrial capacity in aged osteoblast?
Yeah, I don't know the exact number how many mitochondria osteoblasts have, but they definitely decline with age, and you get more reactive oxygen species with aging. That's one of the things that actually induced osteoblast and osteocyte senescence. So there's definitely like a function for mitochondria in aging.
Got it. Okay. So now time is like come, and I feel like today like our members have a lot of like valuable insight from lecture. And as you know, like we have so many questions. I really appreciate everyone's like discussion, and thank you so much for your efforts, like Naomi. And I also like learned a lot. And Next seminar, we will skip the December, and we will have a lecture from Professor Frank Gore, Rush University, at January 26th. So I hope like we will meet there on January. Thank you so